Okay, so in this lesson, we're going to be spending a little bit of time talking about how some substances move across the membrane. Unlike water, they actually have to be sort of pushed across the membrane. So we've been talking about water a bit, and water can move through the membrane without any help. It doesn't require any energy, but some other molecules do need help to move across that membrane. Now, in some cases, these are molecules that would ordinarily move in and out of the cell, but proteins help transport them. We call this protein-mediated transport. We already talked about the structure of the membrane, and you know that there are some proteins that span the membrane and actually connect the inside and outside of the cell. In this case, the proteins act as channels, and things can pass through the middle of the protein, really. In this case, proteins are dynamic, okay, similar to enzymes. They interact with the molecules that they transport. So the molecule will come in, it will interact somewhat with the protein, and then it'll be sort of pushed through or assisted through the membrane. Now, another word for this is passive transport. This is the free movement of molecules through proteins. So these are substances that are moving down their own concentration gradient, similar to how water moves down a concentration gradient. These are moving down their own concentration gradient by diffusion, doesn't require energy. In this case, the proteins are kind of like pores, or you can think of them as like gates or doorways that are open. There's no energy needed by the cell, and the molecules can move both directions. So this just provides a sort of a spot for substances to come through that normally couldn't just come through the membrane. So if we look at this graphic, this is the lipid bilayer of the membrane. Here we see the concentration gradient goes in this direction, and we have a couple of different things going on here. We have simple diffusion here, which would be a small ion, I'm sorry, not an ion, a small molecule like oxygen or carbon dioxide or water. And then we have a channel here, which is a protein, and we have what we call carrier-mediated, where the molecule is interacting more with the protein. But in both cases, they're moving down their own concentration gradient. It's called passive transport because no energy is needed by the cell. Then the rest of what I want to talk about this time is about active transport. And this word active indicates that cells have to use energy in order to transport substances in this way. In this case, proteins are involved like they were before, but in this case, they act like pumps. They're actively pumping molecules against the concentration gradient. So this is cells pushing molecules in a direction that they naturally would not be going. And there are a variety of reasons why cells would do that. But remember that active transport will require the use of energy by the cell. So here's our graphic again. You can see again the lipid bilayer of the cell membrane. We have our forms of passive transport here using proteins, but now we also have active transport, which uses energy. It's a similar kind of a protein structure that spans the membrane, but in this case, the direction of motion of this molecule is against the concentration gradient. So these molecules, the molecule that's passing through here, will naturally want to go this direction because of the concentration gradient, and the cell is actively pushing it the other way. It has to use energy for that. Now, the energy that cells use, we haven't talked a lot about this yet, but it comes from this molecule called ATP, or adenosine triphosphate. This is the energy that cells use, and the energy is really contained within a phosphate group bond. So we have this molecule called ADP, adenosine diphosphate, which is adenosine with two phosphates on it. And then when you add that third phosphate, the bond with the third phosphate, so this is triphosphate, three phosphates, that last bond carries a little package of energy with it. And we'll talk more about that probably week after next. But ATP is carrying that energy. When the energy is added, it changes the shape of the protein. When the protein changes shape, the channels can push the molecule against the concentration gradient. These channels are only one way, unlike the passive transport channels where the molecules can go both ways. In this case, it's being pushed one direction only. and they're used to maintain concentration gradients that are needed by the cell. So sometimes the cell wants to maintain a high concentration somewhere, and it will do that by actively pushing the molecules. This is one example. This is called the sodium-potassium pump, and this is in nerve cells, and it's extremely important. We'll talk more about it later in the year, but you can see here in step A, we have 3 sodium ions really, come in. They're going to be pushed out of the cell. They come in and bind with this protein here. At the same time, 2 potassium ions are coming in from outside the cell, and they're going to bond in that place. When they do, the sodiums are released. Okay, so now the potassiums come in, and then they're released. And then this one protein is really going back and forth, pushing sodiums this way and potassiums this way. And what the cell's doing is increasing the concentration of sodium outside the cell and increasing concentration of potassium inside the cell. And it does that on purpose, but in this case it's pushing ions. All right, another form of active transport doesn't involve proteins, but we call it bulk transport because it's big amounts or large particles that are being transported. This is for very large molecules like big proteins, or sometimes whole organisms. And this is the use of vesicles to move large amounts of stuff. In the first form, it's called exocytosis. Exo means out, and cyto is cell. So this is a process of pushing something out of the cell. We call this excretion if it's a waste product, or secretion if it's a product that can be used elsewhere. And this is where a vesicle releases its contents outside of the cell. There are sort of 2 ways that cells handle this. There is constitutive or calcium-independent exocytosis, and in this case, it's just unregulated, constant removal of substances coming from, it looks like the Golgi apparatus here, packaged into vesicles and then secreted. There's another form that's called calcium-dependent or regulated exocytosis, and this is a very specific case. In fact, this is, again, in nerve cells, where there needs to be a signal for the release of these substances. In this case, it's secretion of some specialized molecules. In this case, calcium is required. So when calcium moves into the cell, and of course that's going to be protein mediated, that's going to be active transport by protein pumped in, the calcium coming in actually causes the exocytosis. It causes this vesicle to fuse and release its contents outside the cell. So that would be a regulated by calcium process. This one is independent of any kind of regulation. There are 3 different ways that cells take in vesicles, take in molecules through vesicles. The first we just call endocytosis. This is where an indentation pinches off and forms a vesicle inside the cell. A very specific case of endocytosis is called penocytosis. Peno means to drink, and this is where some fluid is taken into a vesicle, and the fluid might contain a variety of other molecules that the cell wants, amino acids or small carbohydrates or something like that. And then there's phagocytosis, and only certain specific cells do this, namely a cell like an amoeba or a cell like a white blood cell. Some kinds of white blood cells do this. This is where whole organisms, like a small bacteria or something, or cellular debris, dead cells, parts of dead cells, are brought into the cell, and the purpose is to break them down or destroy them. So white blood cells will take in whole bacteria and destroy them through this process, and this is called phagocytosis. Phago means to eat, so this is eating. Penocytosis is to drink, and then endo simply means inside. Endo is inside the cell. So here's a diagram showing the 3 of these. In the first example, phagocytosis, we have a solid particle, or this might be a whole organism, and it's taken in through a vesicle, and there's a vesicle forming around it, and it's going to be digested. It'll probably fuse with a lysosome, and this thing will be digested. In penocytosis, a little sac of fluid is just pinched off, and within there you'll have some molecules or particles of some kind that the cell wants. And then there's just plain endocytosis. In this case, sometimes you'll see a certain process takes place where protein molecules of certain kinds will come along and they will begin to bind with the cell membrane. When they do that, it causes a pinching off of the cell membrane. In this case, these particular molecules that the cell is taking in are binding to these, they're called receptor proteins, and then the vesicle pinches off, and then they've taken in, now the cell's taken in these molecules, these particles that it wanted. So that's it for this lesson, and this is mainly we've sort of covered everything having to do with the cell membrane. There's a couple more topics we're going to be talking about, and you guys are going to get to do your projects with that. So that's it for this one.